So during active inflammatory bowel disease, cells of the inflamed gut can release their contents into the surrounding tissue and circulation, where they can be recognized by the immune system, leading to increased inflammation and are termed danger signals. One such danger signal we are particularly interested in is mitochondrial formulated peptides. So all cells have multiple mitochondria, and these act as the powerhouse, producing energy for our cells. And interestingly, it is thought that mitochondria descended from bacteria over 2 billion years ago, and this is due to them sharing many similarities with bacteria, such as its formulated peptides that can be recognized by our immune system, leading to increased inflammation within the gut and ultimately resulting in tissue damage. And so it has been previously found that there are high levels of mitochondrial formulated peptides within the blood of IBD patients, and we are now interested into how these can stimulate an inflammatory response on a specific immune cell type called neutrophils. And these neutrophils are found in high numbers throughout the gut, not only fighting off bacteria and infection, but unfortunately leading to increased damage of the gut lining. So what our team does on a regular basis is we receive whole blood from a patient and we spin it at extremely high speeds to separate out certain components of the blood. Due to the specific weight and size of each cell type, your red blood cells collect at the bottom, a layer of immune cells or white blood cells collect in the middle section, and these include neutrophils, and at the top section, this is your plasma, which contains a whole host of different molecules, including mitochondrial formulated peptides. And so firstly what we do is we test how neutrophils react to danger signals, and so in this case formulated peptides. So we add formulated peptides to neutrophils, and we look at how this stimulates an immune response, and we found that formulated peptides significantly increase neutrophil activation. Secondly, we test how we can block the inflammatory response with a known drug. By adding drug A to neutrophils along with formulated peptides, we could significantly reduce neutrophil activation following formulated peptide stimulation. And so our research is simply split into two main aims. Firstly, can these mitochondrial formulated peptides be used as a biomarker to detect or measure inflammation in IBD? And secondly, can we identify a new treatment that could block this inflammatory process to reduce inflammation within IBD? But most importantly, we are interested in hearing from IBD patients on how to better improve our research. For example, how do you feel about donating samples to research? For example, immune cells, so that we can investigate different inflammatory pathways, or tissue samples, so that we can look into providing a more personalised treatment approach for IBD. And additionally, would patients like to be more directly involved at the scientific level of our work? Thank you all for listening, and please feel free to contact me at millie.mcallister@ed.ac.uk.